mentioned, we've done our PhD together in semiconductor manufacturing. So that's my background, really microengineering and microtechnology. But then over the years, I've migrated onto how to use microengineering when we need to apply these devices, these systems, to biological targets. And this is what you see, for example, here in the image in the center, where you see a model of the brain, and you see an electronic device laid on top of that. So more to that in a few slides. So I'm going to skip this introduction about myself, since Jerry has provided enough information. Just one mention is what I'm showing today is mostly what I've been doing during my years at EPFL in Switzerland at the Center for Neuroprosthetics. And really, it was a nice environment because it was a convergence of very diverse expertise. So we had people from microtechnology, such as myself, collaborating closely with people from neuroscience, people from imaging, and translational animal platforms, and also hospitals around the area. And the way that worked, by the way, was that management somewhere high up decided to shove all of us all together in the same brand new building. So we were forced to talk to each other. So that collaboration would be encouraged. Okay. What can soft bioelectronics do for us? So I'm going to show you a video here. You see a rat that is suffering from paralysis, and it's actually held over a treadmill. So you can see that the rat cannot move the limbs because it's suffering from paralysis. But then if we apply a soft bioelectronic implant onto the spinal cord, we can apply electrical stimulation, and this can actually recreate artificially movement in the limbs of the animal. So you can see that now we can artificially reproduce the locomotion intention. So this was what we did in the lab, and then our colleagues at the hospital applied the same therapy to a previously paralyzed patient, and through rehab, he was able to regain locomotor activity, as you can see in that short video. So in short, bioelectronics are electronic circuits or devices that establish dialogue with biological tissues or systems, and this can be used both to sense, so to detect biological activity, or to actuate, so to perform some function. So this is what you can see in this example here. When we talk about soft bioelectronics, what we mean is that we manufacture these devices using soft materials. So we leverage the mechanical advantage of soft materials to enable long-term integration with biological systems. So if we focus on the electronic part of soft bioelectronics, what we leverage is the intrinsic electrical wiring that we have within the body, especially in the central and peripheral nervous system. So the brain, spinal cord, and peripheral nerves are composed preliminary by neurons that are cells that actually establish signaling pathways all over the body, from and to the brain and to peripheral organs. So we can actually leverage this to our advantage because we know that there are signals traveling back and forth. So we can intercept this activity, so we can detect, but we can also elicit this activity by means of electrical stimulation. So we can actually go and apply artificial signals to help the body perform several actions. So this is a more realistic picture of what an ensemble of neurons would look like. So there's a massive population of these neurons, and then we come with some device that is placed in specific locations to then help us perform the function that we need. In the case that I showed before, this is what we use. So we have an implanted electrode array. So you see the image of the device on the top. So it's from an electronic point of view, it's a really basic device. There's just traces of conductors, thin film, that are then routed onto the soft substrate, and then they end up in electrodes that interface with the tissue. So the tissue in this case was the spinal cord, and you see that in the diagram, in the cross-section diagram on the bottom. So you see that curved cross-section of our device here with electrodes interfacing specific locations on the spinal cord. So the idea was to apply current pulses to the electrodes to activate the motor neurons that are populating that region of the spinal cord. These neurons, they go and innervate specific muscles in the body that then are activated by means of our electrical stimulation. So we apply a current, and in response, we get the contraction of some muscle pools. And this is how we were able to reproduce the locomotion activity. What are the challenges? When we think about electronics, we're used to thinking of circuits. Some of you may be thinking of PCBs. Some of you may be thinking integrated circuits on wafers. In any case, we're used to thinking of very planar geometries. So we make devices, we make electronic circuits flat on surfaces. When we think about the body, we're looking at complex topologies. So we have very curved, very convoluted geometries, and also we have mobile bodies. So the body is not a surface that does not provide any surface that is static. We move, and also if we don't move inside of us, there are organs that move physiologically. So if you want to have a more scientific look at this, there is a material property that's called the Young's modulus. The Young's modulus measures the rigidity or the reluctance of a material to deform. And when we think about electronics, we're thinking mostly about materials of high rigidity, so high Young's modulus, so typically metals and oxides and silicon. In the arrow that you see here on the diagram, that's a map, qualitative map of the Young's modulus. So on the far left end of the arrow, we have Young's modulus in the order of hundreds of gigapascals. And this is where conventional electronic materials sit. Now, if we look at biology, most of the tissues in the body, with the exception of bones, but specifically if we want to talk about the neural interfacing, the nervous system, is composed by tissues that are very soft. The Young's modulus has been characterized in the range of hundreds of pascals to tens of kilopascals. So you can see there's several orders of magnitude difference between the materials that we use for electronics and the tissues, the surfaces and the materials that we have to interface with inside the body. So the goal of soft bioelectronics is really to transfer electronic function from these conventional materials, so silicon, to materials that are mechanically more resemblant of what we find in the body. And so if we look at the images there along the arrow on the top of the slide, you see some examples of this. So we see electrodes manufactured on polyimide. Polyimide is a polymer that has Young's modulus in the order of gigapascals, so significantly less than silicon, but still very far from the biological tissues. And the step forward would be to use silicon, which is what we used in the study I showed before. So it's a silicon elastomer, it's rubber, and it's stretchable. So the engineering challenge was how to make devices on top of this material. So we'll come to that. Additionally, we've talked about how the body is not static. So you see there an image, an MRI image of how the brain naturally pulsates. So if you want to implant a device into the brain to perform some neuroprosthetic function, we have to make sure that the device is able to accommodate this natural movement that you see in the clip here. So what happens when we implant conventional electronics into soft tissue? In other words, why do we bother doing this? So here you see a representation of a shank, what we call a shank. You see this device pointy device in the middle here, inserted into the brain. These blue and red cells here are neurons. So when we insert such a rigid device into the brain, we have an adverse reaction that we call foreign body reaction. In other words, the body detects that a foreign object is disrupting the natural order of the cells and reacts to that by activating an immune reaction. So this both destroys the tissue, because we're damaging the tissue, of course, but also destroys the device, because the body reacts to the presence of this foreign body and activates a glial scar. And what happens is that the device becomes encapsulated by scar tissue. And when that happens, basically the device is isolated from the body and we cannot interface with the body anymore. So this is why we want to use soft materials. When we use soft materials, these are more transparent to the body, in the sense that the body is more prone to accepting the presence of such devices in the vicinity of cells. Therefore, the immune reaction, the foreign body reaction, is mitigated, and we can therefore use the device for much longer periods of time, enabling long-term therapy. What do we need to manufacture such soft bioelectronic devices? So of course we need soft substrate materials that in addition have to be biocompatible. So biocompatible means that they're not toxic to the body, and also mechanically they can accommodate the natural body movement. So we need devices that are elastic, in other words. To perform electrical function on these substrates, we obviously need conductors that are able to sustain such deformation. So we need stretchable conductors. And then we also need electronic devices that are embedded into the system. In the case that I showed before, this was, we used electrodes. So electrodes that are interfacing with the tissue to perform electrical stimulation. So when we put all these ingredients together, then we can manufacture conformable and stretchable electronic circuits that we can use for biointerfacing. You see in the example there on the bottom right, one of these devices that is placed on the surface of a model of a brain. You see the surface of the brain is not flat, but it's very convoluted and irregular. But if we use soft materials, like in this case, we are actually able to conform to the entire surface and interface the entirety of the area that we want to address. Now, two important questions. So first of all, we are assuming that if we use soft materials, so soft interfaces, we can get better biointegration. Yeah? We can achieve conformability, and the body can absorb or tolerate the presence of such devices. The second question is, how can we engineer electronic function of these soft materials? In the interest of time, I will skip over a few sections here. I'll just show you one example here. This is how we proved that using soft materials actually is a good idea for the body. So we did this experiment in rodents. So the three diagrams that you see are the central nervous system of rats. So we had three populations of rats. One was sham operated. Sham operated just means that they received surgeries as if we were implanting a device, but we didn't implant any device. So it's just a control group to check what happens just due to the surgical procedure itself, without any presence of material inside. The center group were a population of rats that were implanted with a device that was actually manufactured with stiff materials. And the right group is a group of rats that were implanted with one of our devices made with soft materials. The devices were kept in for 6 weeks, and then the animals were sacrificed, and then we looked at the tissue response in these three groups. And these were the results. So if you see the 3D reconstructions in the sham operated, so the animals that did not, that received the surgical procedures but no implantation, you see that the tissue stays healthy and intact. In the center group, you see the soft implant device, so the device that we wanted to test. And you see that the tissue reconstruction looks very similar to the sham operated, meaning that the device did not affect significantly the tissue that we wanted to work with. And the third group, so the stiff implant, the population that received a rigid device in the spinal cord, suffered from significant deformation of the tissue, which then causes a lot of problems, locomotion impairment, and not only. So this convinced us that, yeah, this is the way to go. We need to use soft materials. But then we have the second question, which is, okay, we have the materials, we have the silicone rubber. So how do we make conductors on this substrate that are able to withstand the stretching and the deformation? And so we talk about how to engineer elasticity in materials that would not be normally elastic, and therefore we talk about stretchable conductors. Now, by means of abstraction here, if you think of a log of wood and you make it thin, 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 imagine having a sheet of paper. Now, obviously that's not the case, but just bear with me for a second. If you have a thin sheet of paper, that's obviously flexible. So you can flex your sheet of paper, although it comes from a material that is not really flexible. If we go beyond this step, on top of having things that are thin, we also make some cuts into this material. So we introduce some sort of structuring in the material. We can make it elastic. And this is what you see in the image in the bottom left. So this is a sheet of plastic that is flexible because it's thin, but it would not be elastic normally. But because we performed some cuts in the material, we can now elongate it as we please. This is the strategy that we used to engineer elasticity in thin film metals. So we can deposit thin films of metals up to hundreds nanometers on this soft elastomer substrates. But then we also engineer cuts onto the surface of these materials so that we can actually elongate them. And this is what you see in the microscope image on the bottom right. So you can see these cracks in black. They act as mechanical buffers. So imagine this is a film of gold on top of a piece of rubber. So you can elongate the piece of rubber. The rubber is elastic, so it will elongate, of course. But now what happens to the gold is that because we have these cracks in the material, these cracks can expand. So when we elongate the rubber, the cracks in the gold expand, and the areas that are not cracked in the gold, they stay conductive. So we always have a conductive path connecting all the solid islands in the thin film gold. And if you see that in practice, this is what it looks like. So we have a device that is thin film gold on an elastomer substrate that we can stretch as we please. And this will always maintain electrical conductivity. So we can use this technology then to manufacture complete devices. This is one of the devices that we used in that study. You see that we can stretch it. In this case, we're stretching it by hand. You see also a droplet coming out of this device. This is because we also embedded some microfluidic device channel on top of that to actually deliver some pharmacological agents on top of the electrical stimulation. Okay. So what we did with the gold was actually tune the fabrication process so that this actually happens naturally in the gold. In other words, we have a machine that deposits gold on the rubber. If we tune in some certain way the conditions of this deposition, then the gold grows or is deposited in this with this microscopic structure. But gold is not really a good material for medical devices because it tends to dissolve and corrode in the body. So we wanted to have the same approach, but with other materials. But we needed in a way to be actively introducing this microstructuring patterns, and we can do that with microfabrication. So we can use a technique that is called lithography, where we actually design in a CAD file the patterns that we want to transfer then into any material, and then we're able to do that by means of several technological processes. This is done in a clean room environment. So I have a short movie here, courtesy of the clean room where we did this work. So we have several semiconductor processing capabilities there, but we're really tuned to for use on polymer materials and soft materials. So this is one of the devices being manufactured. So when we take control of the microstructuring properties, we're not just letting the materials deposit with a random cracked pattern, but we're actually introducing a really precise structure at the microscale that enables us to also fine-tune the mechanical properties and electrical properties of these films. So in the movie in the center there, you see one experiment where we were actually testing the durability under stretching of these materials. And in the bottom example, we applied that to films of what's called ITO, so indium tin oxide. Indium tin oxide is an interesting material for biological applications because it's a conductor, but it's also transparent, which is a rare combination. So we can have devices that have conductive traces, but still we can look through them, so we can image the tissue underneath. So we can apply this microstructuring technique to that, and we can stretch it and measure the resistance, and also do some reliability measurements. So just look at what the endurance of these devices would be. Another approach that we tried to introducing elasticity in soft devices was to use liquid metals. So liquids are obviously intrinsically stretchable because they're liquids, but how do we confine them to the places where we want the electrical conductors to be? This is what we came up with. Well, not we, but my colleagues. I was not fully involved with this, but I can tell you in a few words. We had our silicone substrate, so the elastomer that we work with is called PDMS. We applied a thin film of gold again, but the gold in this time is not stretchable because we want a thicker metallization. So if you grow the gold too thick, it will not have this characteristic microcracked structure. So to improve on that, we wanted to see whether we can use the liquid metals to help us. So what we did was to deposit gallium. So gallium is a metal that is liquid at room temperature. So this is quite interesting because we can have a metal that is liquid around the temperatures that we have in the body. If we deposit gold and then gallium on top of the gold, then we have this formation of what we found to be biphasic liquid-solid conductors. In other words, we have the gold that acts as a solid base onto the PDMS, and then the gallium on top that acts as a liquid parallel conductor. When you stretch this device, what happens is that the gold will crack, so you will have severed electrical connections. But the gallium on top will actually bridge these severed connections because it stays liquid, and then it flows wherever you expand the device. So this was another way of introducing stretchability into soft devices. And I'll just show you a quick demonstration of the use of this. This is a PDMS membrane with liquid metal tracks. And you can see that we can inflate with a pump this membrane. All the while, you see all the electrical connections on the membrane, they stay functional. So you see all the LEDs being lit, going through all the stages of deformation of the membrane. Okay, so to the more interesting part, I think. What can we do with this? So I've shown an example of implantable software electronics. So we've used that for soft spinal cord stimulation electrodes. We use that. I've shown that for locomotor restoring, neuroprosthetic use. We've also used that for regulating the blood pressure. So if we, if we stimulate different regions of the spinal cord, we can activate different neural circuitries. So in the rat, in the first clip that I showed, we were activating the lumbar motor circuitry that innervates the muscles so we can actually perform locomotion. If we implant the same device in the thoracic part of the spinal cord, then we're actually interfacing with what we call autonomic neural circuits. And these regulate, amongst other things, the blood pressure. So we're actually able to, by fine-tuning the stimulation parameters, so the current, amplitude, the frequency, etc., to regulate the blood pressure in animals that were suffering from a decreased blood pressure. We've also used that for upper limb restoration of movement, so for reach and grasp action in primates. If we change completely gears, we go into wearable devices. We can also use these, we can also apply these devices not inside the body, but outside the body. And we can use these as electronic skin sensors. And I'll show an example shortly after this. Here you go. So we made devices that were very similar to the membrane with the LEDs that I showed before. And the layout of the device looks like what you see there on the left. So you see the hand, and you have devices with serpentines laid on the joints of the hand. So these serpentines, they act like strain gauges. So you can imagine that if you stretch this device, the resistance of these conductors will change, and you can detect that. Now, if you apply this to the joints of the hand, when you move the hand, this will automatically stretch the device, and therefore will produce a change in the resistance of these conductors. So we use this in several different ways. One way that we used this was to help neurologists assess the entity or the progression of a Parkinsonian tremor. So as you may know, Parkinsonian patients suffer from trembling, so they will have a very unsteady movement in the hands. So up to now, the way this is assessed in the clinic is by having a qualitative form, or assessment form that the physician fills at every periodic visit. But it's based on subjective observation of the patient by means of the physician. Here, we're actually able to introduce really quantitative measurements of that. And you see the example of the traces on the top right plots. Here, what you see is the resistance of one of these devices on one of the joints in a Parkinsonian patient as the time goes. So you can see all the tremor going up and down, moving up and down the resistance of the device. So you can use that to actually monitor progressively over time how the disease is progressing. Another application for this was actually to track the movement of the hand in healthy subjects. So for virtual reality applications. So what we found is that if we have enough of these sensors on the hand, we're actually able to distinguish different actions that are performed by the hand. So we had a power grip performed by one of our volunteers. Power grip just means grabbing an object and applying force in it. The pinch, catching a bottle, etc. And so these are plots that demonstrate that it is possible to do, but we went beyond that. We actually built a model of this. You see the volunteer moving the hand, and we're actually using the signals, so the change in resistance that we detect, to drive a virtual reality model of the hand. So you can see there's a pretty striking representation of the hand. So you can see the hand has moved, has rotated, and the model has not rotated because we don't have any sensor detecting the rotation of the wrist. But if you look at the fingers, this is super accurate. So we are quite pleased with that. So I'll move to the conclusions. So the goal for me today was to give a very brief overview of my field of research, which is how to apply microtechnology and soft materials in combination to solve challenges that are really relevant today in the clinic. So for medical research, with stray applications that go beyond clinical research. For example, we've seen the virtual reality model. This you can very well imagine this being applied to gaming and other fields. I am driven by challenges that arise in biology. So what I do is driven by the problems that biology gives us. And our goal as engineers and researchers is to actually find solutions that are able to accommodate the needs of patients or biological systems in general. And the reason why I like this is that it's very multidisciplinary. Obviously, I've shown things from several different collaborations that I've had. There's a lot of people who have contributed to this, coming from very different expertise. So the engineers, of course, also the neuroscientists, biomedical imaging experts, clinicians, etc. And what I found over the years is that there's really a language barrier between different disciplines. So it's been challenging, but also very rewarding to actually work towards breaking some of these barriers and actually making collaborations more of a norm rather than auditing. And I hope this sparks some interest in this field of research. And I'd like to thank you for your attention. And if you have questions, I'm here to try to answer most of them, all of them.
Thank you. Thank you. This is very exciting. Quite a different topic for the team. It's very engineering, and I think everybody has some engineering in them. Yeah. So I'll start by very with a simple question. So you were talking about metals layered on top of on plastics. So what technique is used to deposit like gold on top of plastic?
So we have different techniques. In this example here, with the spinal cord electrodes, this one, this was thermal evaporation. So basically, for those of you who don't know, you just take a target of material, so gold, you place it in a vacuum chamber and you heat it up to its melting temperature, and then you have vapor of gold raising up in the chamber and then hitting your substrate that is placed above. For the liquid solid conductors, the gold was spattered. So it's a different technique where we actually bombard a solid target of gold with the ions in a plasma, and then this is left to diffuse into the chamber and onto the substrate. Same thing for the gallium. And in this case, for the indium tin oxide, we also use spattering.
Thank you. Harold? You're on mute, Harold.
Thank you very much, and very interesting presentation, and good to see you again. So you know our market is basically to help elderly live longer where they in their homes and stuff. So I understand that the technology and the research you're working on can be applied at some stage also to those. How far away are we before we can put a little implant into a grandma and have her taken better care of?
Yes, so that's $1 million question, obviously. when you work with the medical devices, especially class 3 medical devices, so the ones that are really risky and invasive, the time to market is really long. We're talking about 1 decade at least. The technology that I've shown, so the gold, the thin film gold on elastomer, is actually currently on a clinical trial. So we've gone a long way towards that goal. But anything that you need to start from anew has to go through a lot of validation steps. So you need to build a documentation. You have to do. I mean, things are changing now, but traditionally you would have to do animal tests, collect a lot of preclinical data, and convince the authorities to go for a first in-human clinical trial. And that doesn't mean that you can put implants in patients. That means you can try some version of the implant on a limited population of patients. So from there to actually having that available to people, it's a decade, I would say.
Mm.
Thank you. So not tomorrow.
No, but it also that's why I wanted to stress here that these technologies are good also for wearable devices. This obviously
Yes.
that's a completely different landscape in terms of approval. So we were able to apply one of these devices onto Parkinsonian patients right away. We didn't have to apply for any authorization. So it's really 2 different worlds.
That's a very good. The wearable aspect, I think, is a much quicker way to get in the market with your technology. But the same kind of techniques that you're doing for implants, but it's a way to validate as well. Yeah, it's very interesting how it's like something wearable on the hand, and also now with new AI and imaging, videos can also do very similar tracking of the hand and all. But then you need a camera and all this, right? So yeah, it's quite cool to see some accurate measurements coming out of it.
It was quite good.
Yep.
Yeah.
Yes, so thank you for the presentation. It was very intuitive and very thoughtful. What my question is that all this research is composed on the physical impact. Like we are talking about installing something on a human brain or a human surface, getting in contact with the neural system. So all this is like the contact version of it, like the physical version of it. So is there some physical blockers with the wireless implementation of these things? Like a few times before, there was some serious research in the wireless communication with the neural systems. So what do you think? Why is this a way more resourceful or more like progressive?
So that's a good question. So wireless does come into play in this. So let me take some time to clarify here. The reason why we want to go inside the body is not because we like to to open people. It's because there's so many neurons in the brain, for example, that if you go too far from the actual population you want to detect, the signals will mix with signals coming from other populations of neurons. So what you have if you go far away from the surface of the brain is what we call compound brain activity, and you lose the specificity of the signals coming from a precise region of the tissue. Now, it depends on the application. For example, you see electroencephalography devices. These are things that you put on the scalp, and then you have electrodes, and you can measure the brain activity, no problem. But if you want to do neuroprosthetic therapy, you really need to be able to detect signals coming from regions of the brain that are spaced apart millimeters, if not less. Sub-millimeter. If you are too far from the surface of the brain, you will not be able to distinguish the signals coming from point A and point B, if point A and point B are spaced only a few 100 microns. That's why we really need to go inside the body. So we cannot really use wireless interfaces for that. What we want to use wireless is once we detect the signals from the source, so from close to the neuron, we can reroute them to some electronic box, whatever, and this is placed somewhere subcutaneously, so under the skin. And then we really don't want to come out with big cables or many cables. That's really a problem in the clinic today, and this is why we're limited to only few channels for neural recording. So if we had the ability to route hundreds of signals from precise locations of the brain onto a collection box, and that has a wireless transmitter, and then we can collect all the data wirelessly, we'll be very happy. So there are examples of this today, but there are limitations also. The bandwidth, the amount of data that needs to be taken out is quite huge. So I'm not an expert in data or signal transmission, but I know there are really significant limitations in that, and there's a lot of research trying to overcome these limitations.
Thank you so much.
Thank you. Jahin.
Hi, Giuseppe. Thank you for your presentation. It was very interesting. So I've got a couple of questions. Firstly, the human test subject that you used on humans, is it possible that some kind of this kind of technology can be evolved to give therapy and eventually make the normal nervous system impulses going back again so that they can get up from paralysis?
Yeah, that's a very good point. Actually, this is probably I wasn't very clear, but this result here with the guy walking there, he doesn't have the system inside anymore. So he used the system for 6 months and used it as a therapy system. So by using this artificial restoration of motion, then you leverage what you call neuroplasticity. So the nervous tissue is able to rewire itself, and the spared circuitry that remain functional in the body, we're actually able to retake some of the lost function. So yeah, that's the goal. That's the goal. There are limitations. Let me add. If you want to do that, to date, it's been successful only in patients that have had some spare activity. Otherwise, for people that have complete paraplegia, for example, that's not working. So you would have to use the system as an artificial assist for locomotion.
Giuseppe, I just want to remind you of the time. Do you have to leave?
I can stay a few more minutes.
A few more minutes, okay. Like a very quick couple of more questions, okay? Arslan?
Thank you very much for sharing such interesting information with us. Actually, I don't have a question. I just wanted to know your point of view. Actually, a few years back, around 3.5 years back, I attended a session on implanting and device inside the human brain. The speaker was Dr. Naveed Imad Sayed. So he actually told in the presentation that their team actually successfully implant a semiconductor device inside the brain of a mouse, and then they train a mouse to actually switch or turn off some devices. And another topic was actually also of his research was to study the human behavior of patients having epilepsy. So I just wanted to know that what is actually the current update on those fields, like.
So there's two things that you mentioned. The first thing is implanting a device in a mouse brain and doing some training. I think the most advanced demonstration that you have, I mean, you've seen maybe the Neuralink marketing material. So Elon Musk's venture into neurotechnology, but basically we can implant these devices successfully in people. That's not the limitation. I think the limitation is But there's two limitations that are currently overwhelming. First is the devices, if they're not biocompatible enough, they tend to fail over time. So we have patients implanted with the electrodes in the brain, but after a few years, you start seeing devices failing. And what you really don't want to do is tell this patient, okay, we need to have another surgery because we need to replace your device. No, you really want to have a device that lasts decades, if not more, a lifetime, ideally. And this is why research in materials is very important. The other limitation is if we admit that we can implant safely these advanced devices with loads of channels, we can do a lot of recordings. We don't know exactly what to do with this many channels, with the data. So we have loads of data, we don't know what it means. So it doesn't mean that we don't know how to treat the data. It means that there's a lack of fundamental understanding of the neurobiological mechanisms, really. Because if, for the first time now, we have the opportunity of studying this many signals, it's still new to us. We don't know what they mean. So we were able to do basic functions, which are already very good. For example, locomotion, we know how to reproduce that. We know how to detect that in the brain. But anything that goes beyond that is still challenging. And the second aspect that you mentioned is epilepsy. We are actually working with the epilepsy patients, and there is a very good point, a very good case for our technology, because for drug-resistant epilepsy, what happens today is that the neurology and neurosurgery teams, they meet with the patients and they schedule a surgical operation that is aimed at removing a tiny portion of the brain that is causing these epileptic seizures. How do we know where this region is? Is by having a surgery before the resection surgery, opening the skull, implanting an electrode grid, much like the ones I showed before. Let me pull it up again. So it's this one. But imagine now we're having a clinical grid, which is made with the kind of rigid materials. Because it's made of rigid materials, we cannot keep this grid inside the brain for a long time. So we're limited to 30 days by regulatory norms. We use that over the time of 30 days to actually wait for the next few seizures, epileptic seizures. And we use the data we collect from these electrodes to understand where the epileptic foci, so the source of epilepsy, are in the brain. So once we know that, we do the other surgery, we open again, we remove this electrode, we do the tumor, the tissue resection, and then we close up. But we are very limited by the materials. So we have a very good case here by using this soft technology, because we can insert these devices and leave it for a much longer period of time. We have much finer control on the number of seizures that we can detect, because we're not limited to 30 days. Two, we can microfabricate the electrode placement on the device, so we can actually be very much more selective spatially on the surface, and therefore we can confine the resection region. So we are, really sparing tissue for the next surgery. The third advantage is that if you have an implant of this sort, the clinical type, you are not allowed to go through MRI scans because it's not safe. So the metal in the device will overheat, and it's dangerous for the tissue. When we use thin films, the amount to the mass of the metal is much more limited compared to conventional devices, and therefore the heating of the device is negligible. So therefore, we're actually safely allowed to go into MRI, so we can image the tissue at the same time, which is a great advantage.
Last question, Safi, very quick one.
Thank you.
Yeah. So First of all, thank you so much for the session. My question is basically related to on top of what Aslan just asked. So, for example, there's Neuralink, which actually implants brain chips into the human brain or animal's brain as of now. So, I would like to know, like, how does the device actually interpret some intrusive, unwanted, or irrelevant thoughts? For example, we all have nagging thoughts that we cannot control, right? And let's see that, for example, we can accidentally start playing a song on our phone if the wrong thought crosses our mind. So how does these kind of chips or the Neuralink kind of you know flag those intrusive thoughts?
Yeah, so this is exactly the limitation I mentioned in one of my answers before. It's the first time we are able to record so much data from the brain. We don't know what it means. So the short answer is we don't know. They don't know. Nobody knows yet. I'm sure we will know at some point, but interpreting the data is very difficult. I'll just explain something. There are examples of patients that have received similar implants to those of Neuralink. And they use the signals coming from this implant to actually move a cursor on a screen. You may be familiar with that. The way that works is that you tell the patient to think about doing something, think about performing an action. And while he does that, he or she does that, you record the activity from all the electrodes that you have in the brain. And you do that repeatedly. And this builds you a calibration map. So you know at some point that that pattern of signals coming from the electrodes means that the patient is thinking, I want to do this. And later, after the training and calibration, basically you can tell your system, your computer system, when you detect a pattern of signal that is similar to this template that we have, it means that the patient wants to do that. So therefore, move the cursor this way or that way. But it's very elementary. I mean, it's elementary the way I explained it, but it's not different the way it's done. It's quite elementary. And this works with tens of signals, anywhere from a few to maximum 90. Yeah? Neuralink is implanting electrodes in numbers in thousands. So nobody knows what to do with all this data. They will know at some point, but today they don't know.
I understand. So, like, is there a difference between, like, could be in the neuron activity that are like the thoughts that are intrusive and thoughts that are conscious? So, like, could be some energy change that could actually map the thoughts on it.
So we can't interpret thoughts from electrical signals. What we can do is check where the signals are from, and we just correlate that with known patterns of activity. So at some point, you may ask a patient with one of these devices to think about something, so thought, and check the signals, and think about something else, different colors, and see if there's differences in the signal. But today we don't have that. Today we only have how in different locations of the brain we have signals that mean different activation of different pathways. The easiest is the motor pathways, because you can actually verify that visually. For the rest, it's all in the making.
Thank you. I hope that was very useful for everyone. I enjoyed it as much as you guys did. I would like to thank Giuseppe for taking time to do this. Thank you so much.
No, it was my pleasure.
Yeah, it's very, very good. I think it stimulated our brains as well as to do this. It's quite exciting. Thank you so much.
Okay.
And wish you all the best, and we hopefully speak to you again.
Yes, catch up. And
thank you. Yes.
Everyone have a nice end of the week.
Yes, likewise. Thank you.
Bye. Bye-bye.
Bye-bye. Bye-bye.